Welcome. In this seminar, we are going to introduce computational methodologies for the discovery of bioactive compounds. Drug discovery process is a very complicated methodology and very time-consuming and very costly, involving billions of euros per year and an average estimation of about 15 years. Okay? Since a disease is identified until a drug is released in the market, there are many stages that need to be investigated in order to be able to evolve to a bioactive compound, a drug, and so on. There are three stages, which are validation of a pharmaceutical target, then identification of lead molecules, and finally optimization of this molecule on the evolution to a drug, in which we can help to this project using computational methods. So the main idea of our talk here, of this seminar, is to explain how computational chemistry can help in the acceleration of this project. First, discovery of new drugs. How can we help in this process? Well, let's suppose that we have an enzymatic reaction where a substrate interacts with an enzyme and then some products are released. And let's suppose this is a process that can be involved in a disease. Then our objective will be to regulate or to block or to inhibit this process. How can we do it? Well, there are many different approaches, but here we are going to focus on the introduction of a compound in green color on the slides called drug, which interacts more strongly with the enzyme than the substrate, so that the reaction is not produced at all. The reaction is blocked. Okay? How can we do this? There are many different approaches. One of them is rational drug design. It's not the topic of this seminar, but ligand database screening. We're going to focus now on this technique. On the pharmaceutical industry, ligand database screening consists on very expensive equipment. We are talking about millions of euros, and where target compounds are screened against a very large library of compounds. This is a very precise method, but the main problem is that it is very expensive, so only pharmaceutical companies usually have access to it, and from the other side can be very time consuming. So we propose an alternative to this method, which is based on these three researchers. They received the Nobel Prize of Chemistry 5 years ago due to developments in computational chemistry. Around 40 years ago, they started to apply computers in order to simulate chemical processes involved in very relevant biological processes. So we start from all the results that they obtained. Basically, what we will do is to simulate the high throughput screening methodology, but on the computer. We will process in the computer, sometimes called in silico methods, libraries of compounds in order to predict which compounds will be active or not active against a focused protein target. It is very important to say here that this method, usually called virtual screening, this method can process, here we have some details about the libraries commonly used for such approach. We can have, for instance, public libraries such as Zinc database, which contains millions and millions of commercial compounds, but we can also process in-house generated libraries, chemical synthesis of interesting compounds, or another kind of libraries. But in any case, we need to use high-performance computing. High-performance computing requires processing of lots of compounds and very time-consuming algorithms. So we need access to a supercomputer. Which is a supercomputer? Here we have some idea about it. It's a, let's say, compilation of computing elements. So we can start from a single computer, desktop computer, which can have 4 cores A core is a computing unit, and then we tend to compile all of them until we reach a machine that can have currently millions and millions of cores, and we can perform in parallel millions of operations per second. Also, it is very important to see here, this is a kind of relevant publication, that most of the drugs, many of the drugs that are now on the market and approved by the FDA, Food and Drug Administration, this is the regulatory agency in the United States. Most drugs have been developed by help of a computer, or the computer has played a key role on the development of the compounds. So we are talking about a very mature technology. Okay, we have introduced virtual screening, the need for high-performance computing architectures. Now we are going to focus a bit more on the physical description of the model. Usually, we can model the interaction between a substrate and an enzyme using the lock and key analogy developed more than 100 years ago. In this approach, we can suppose that the enzyme takes the shape of a lock, and our molecule, our substrate, is a key that fits completely into the lock, so that it can open or close totally such lock. So in our approach, we have a protein enzyme that we want to block. Let's say this is our protein target, so we will model it as a lock, and then we need to screen a library, a database that contains millions of ligands, in our analogy keys, and we will predict which is the key that opens or closes, it depends on the case, the most this lock. This is very similar to when we try to find the needle in the haystack. So this is a very complicated process, but it works sometimes. Methods for virtual screening. There are many different computational methods. Here we have a list, some classification regarding accuracy. So pharmacophore methods can usually predict only the pose, I mean the way the key fits into the lock and the score. Then docking methods are a bit more precise. Then we can go for molecular dynamic simulation, and in this case we can add the time dimension, so we can study also how the system evolves in time. And finally, we can have very accurate affinity prediction using quantum mechanical calculations. Well, if we could freely choose one of these methods, we would of course go for quantum mechanical calculations. But there is a problem here, because accuracy goes in the opposite direction to cost, which means that post-prediction methods by pharmacophore methodologies can be run on a desktop computer. They are very cheap. Then molecular dynamics and docking methods require lots of CPUs, GPUs, which means a medium-size or big-size supercomputer, but quantum mechanical calculations are really costly, and even nowadays cannot be applied routinely to the drug discovery process. So what we usually do is that we start to process a library of millions of compounds, starting from pharmacophore methods. Then we make a selection from these 1 million of compounds to, let's say, 1,000 of compounds, and then we apply docking methods, then molecular dynamics, and then we refine a very short list of compounds, typically tens, sometimes it can be hundreds, with quantum mechanical calculations. So this is the funnel approach. Okay, then we can classify virtual screening methodologies also regarding the information, the available information that we have at hand. We mean, on the right side, there can be drug discovery projects where we can have only the information about the protein target or the lock, and then we use the so-named structure-based methods. protein ligand docking, molecular dynamics, and so on and so forth. But from the other side, it is very likely that in most relevant drug discovery projects, there is no crystallographic structure available for the protein targets, but we have only some known bioactive compounds that have some desirable effect on this disease. And then we will try to develop models based on these ligand structures. So these are the so-called ligand-based methods, and then we have similarity searching, pharmacophore mapping, machine learning methods. Okay, these methods are usually a bit different, the ideas, the way in which they are applied to the problem, but usually the best approach is to apply both of them at the same time. Now we are going to focus mostly on docking methods. Docking methods, what they usually do is that they predict the structure, the 3D structure that the ligand adopts when it docks into the active site on the enzyme. Here we can see a two-dimensional description of the interactions between the ligand and biotin and the protein streptavidin, and we can see here mainly having a look at the lines and the dashes, hydrogen bonds, hydrophobic interactions, because docking methods usually measure or model non-covalent interactions such as electrostatics, van der Waals, hydrogen bonds, and so on and so forth. But other more exotic interactions are not usually well modeled with docking methods, like aromatic interactions, metals, and of course covalent bonds. But as we said before, the precision of docking methods is not one of the most accurate methods, but they are usually very, very, very fast. So our idea is to apply the funnel methodology and apply first pharmacophore methods, then docking, then molecular dynamics, and finally quantum mechanical methods. And here, briefly, a description of the typical potential, or better, we can say scoring functions using docking methods when describing the non-bonded interactions. We can see here double summatoria, also solvation, which is based basically on the computation on the molecular surface. And we want to highlight here that the complication, these are not very complicated mathematical formulae, but they are very costly for the computer. But from the other side, they are very easy to parallelize. So up to now, we have described the main problem, the discovery of bioactive compounds. We have introduced some of the methodologies used in our virtual screening approaches and describe the main methodologies, but from now on, we are going to describe some application cases in order to get the idea about how such methods really work in real scenarios. So this project, Discovery of Novel Anticoagulants, it was a collaboration between our group and the Faculty of Medicine from the University of Murcia. And in this case, for blood coagulation purposes, there is heparin. This is a widely known drug, greedily accepted in the market. And we wanted to develop a new anticoagulant molecule, so we need to work on the anticoagulant cascade. There are many, many protein targets involved here, so we decided to focus on antithrombin. Antithrombin is a protein, a structural protein that interacts with heparin. Both are now on the slide. And well, heparin, it interacts, it needs usually 5 interaction sites with the antithrombin, but the problem is that in mutants of the protein, in mutants of antithrombin, interaction between heparin and the protein is partially, and in some cases, totally lost. So in such cases, heparin, it doesn't work. So we wanted to find a novel molecule that would overcome these limitations. In addition, heparin has some and the desirable side effects, hemorrhage, thrombocytopenia, and many others. So, what did people usually do in such contexts? So basically, regarding the bibliography, they focus on the structure of heparin, and they try to derive some similar molecules. Well, in some cases, some interesting results, but still the same side effects. So in our approach, we wanted to perform fully blind docking, which means we would screen or process the whole protein surface, the whole surface of antithrombin, and we wanted to see first as a control if heparin after blind docking would find the typical interaction pattern of heparin, which is the case here, because red color means no interaction at all, and blue means the top interaction site. And in addition, the predicted pose for heparin fits very well the crystallographic structure of heparin. So I'm not going to give all the details here, but there is a detail, the main reference about the publication later, so you can have access to all the details. And then we screen totally without any hypothesis the zinc library, a subset containing 13 million of molecules. We didn't want to focus on the structure of heparin, so it was a totally blind screening, and at the end we selected 10 compounds for testing them in the lab and our criteria was to find compounds that had a score of the same order of magnitude than heparin. So finally, and after some tests, we found that using isothermal titration calorimetry and other techniques, that this compound called TMI on the right side of the slide had an interaction constant about 5 times stronger than heparin, which was a very interesting result, and that it needed, we evolved, let's say, from heparin on the left side, which contains 5 carbohydrate rings, to another molecule containing only 1 carbohydrate rings, which is much more smaller and it needs less interaction sites, so we could avoid some of the problems with mutations on the heparin site. You have all the details there for the reference of the publication. Another application example, now it is a bit different, is Ferry disease. We work in this disease. This is a bit different because previously we had a protein and then we have a interacting site very well defined. But in this case, we are going to see how the focus is different. First, I'm going to introduce a bit the disease. In Ferry disease, people have some mutations in a protein called an enzyme alpha glycosidase. When this protein performs its work, it usually goes to the lysosome and interacts with some substrate and everything is going well. But in people with mutations, this protein is misfolded. Misfolded means that the key structure needed for the enzyme to process its substrate is lost. So, typical approaches, there are many approaches here in Ferry disease. One of the approaches is to provide the patient with an enzyme that doesn't have these mutations, which is called enzyme replacement therapy. But this approach is very expensive and it doesn't always work. So another approach is to provide with a molecule called chaperone, which helps the protein to fold correctly. And this is the approach that we are going to tackle in this problem. Usually the state of the art in this field is that in order to develop new chaperones, what people did is the following. Here we have a depiction of the of the natural substrate of alpha-glucosidase, okay? And what people did is to modify the natural substrate in order to add a positive charge in the carbohydrate ring so that electrostatic interaction between one of the key residues from B phase would increase a lot. So at the end, we have this molecule, DJG, and there is a, let's say, 6 orders of magnitude increase in affinity. Well, this molecule now is on the market, it's called migalastat, and it's a very interesting molecule for Fabry disease. And other researchers are following the same strategy and are developing another molecules basically derived from the structure of the natural substrate. This is very interesting, but there is a very important problem here, and is that when we try to develop chaperones that interact with the active site, we are blocking the enzyme, so that the enzyme cannot fully perform its work. Okay, so the approach that we wanted to develop here is to derive a chaperone, a small molecule chaperone, that would interact, but not on the active site, but in another part of the molecule, so the active site would remain free for perform its function. And this is what we did. We can see there, on the middle of the slide, the structure of the protein, the enzyme, and in blue there is the active site, and in pink color some allosteric site that was suggested by our collaborator in this project, which is from the University of Naples. And the approach was to screen a library of compounds in order to predict compounds that would interact very low or with almost no interaction on the active site, but which would have a high number of interactions in this identified allosteric site. In summary, this was the approach that we followed. We processed millions of compounds from zinc database, then we performed 3 shape similarity against the known substrate, and finally we identified 2,6-D-thiopurine or DTP as one of the few allosteric chaperones for Fabry disease. You have there the main reference in +1, and the most interesting result we can see it here. In this picture, this figure, we can see this is an experiment that represents how the protein, the enzyme, is denatured when we increase the temperature. If you have a look at the X, which is just the protein, let's say when we reach a temperature of about 50 degrees, the enzyme is completely unfold and denatured. So we need molecules that increase this interval of temperature. It would mean that these molecules can really stabilize the misfolded structure of the enzyme in cases where patients have mutations. So DJG can stabilize, we can see there, almost 20 degrees. Our compound can stabilize 5 degrees, which is not so great as migalastat, but the most interesting result is that it can be used in synergy with migalastat. So at the end, we increase a total of 5 degrees the temperature interval of migalastat. So this is the main result of this study in summary. And in addition, and this is more important than migalastat, is that our compound DTP does not interfere with enzymatic activity. And this is very important in contrast to migalastat. So in this study, the main conclusion, apart from the compound, is that we should focus sometimes, and depending on the project, not only on the active side of the enzyme, but in another part of the molecule, of the protein. and also that we have identified some allosteric region of the enzyme, and we have predicted some residues that are key for the interactions. But please do not forget that this is a computer prediction and that needs to be validated by structural studies, mostly crystallographic studies. From the other side, DTP is a very interesting compound. It is safe, it's not toxic, and it can also rescue some mutants that do not respond to the therapy with migalastat. So, in conclusion, the methodology developed here is very important. We call it blind docking, and it can predict compounds that interact with some focus parts of the protein and which are not in relationship with active site on the enzyme. And finally, this is the idea, the workflow that we follow in this kind of projects. So basically, we start from virtual screening calculations. We need to set up the system, then we perform the calculations so that we obtain some virtual screening hits. These are computational results and need to be validated or refuted. So then we perform in vitro tests, and if we are lucky, we obtain some hits. Then we perform some in vivo tests on the same, and then we need to analyze the results. Then we need to decide whether we will optimize these hits using chemical synthesis and publish or patent the compounds. But usually we can only, you can see here that in principle we would perform many different cycles, but in academic projects and due to problems with financing, we only perform one cycle. But the interesting thing, the more practical approach, and if we had more financing, we would perform many cycles using the feedback obtained from previous hits. Okay, this is a last application case. Okay, all these application cases are results from our research, and in this case is a bit different from the previous one. In the two previous cases, we performed a virtual screening against a library of commercial compounds. There was no other criteria. We processed millions of compounds, we obtained some hits, okay? But in this project, this is a bit different. This is regarding Zika virus. Zika virus had some, gave some attention two years ago during the Olympic Games in Brazil, and it consists briefly that the Aedes mosquito can give very harmful effects during pregnancy, so that newborns present microcephaly, and this is very severe effect. So our idea, there was an increase, a very important increase on the cases of Zika virus and in microcephaly, was to provide some help in this case. So what we did, at the moment, 2 years ago, there was no, or better, let's say, there were not many crystallographic structures of the Zika virus protein, but we were paying attention, and during June 2016, there was released one of the key structures of one of the proteins that intervenes in the replication process of the virus. So that we choose that structure, and then we perform a virtual screening, but in this case, and we didn't follow the same approach as before. We did not screen a single library of millions of compounds, so that we focus only on the FDA library. FDA library, or DRUGMAN in this case, contains either molecules approved for the Zika virus, molecules approved for the American market, so that we try to reproduce molecules that were used in another diseases and see if they could work here. What is the main advantage? That all these molecules are already very well studied, and they are supposed not to have toxic effects. So in case we would find a hit, it could be applied directly, or we could go directly to clinical trials, so that the gap between discovery and application would be enormously reduced. So we perform virtual screening of drug bank library. So from 8,000 molecules, we select the 100 most promising hits in silico hits, and then our colleagues from Hong Kong University perform a primary screening. They look into the most favorable clinically approved drugs from our hits, and then we found on the left we can see the in vitro results. The most potent candidate molecule was novobiocin. We can see its structure there, and on the left side we can see that if we increase concentration, it has an IC50 micromolar. So in vitro, the compound inhibits the replication of the virus. It was very interesting. And then our collaborators perform studies in vivo. So in mice, there is a Zika model for mice. And at the end, on the right side, we can see a figure. On the y-axis, we can see a viral load in logarithmic scale. And on the x-axis, we can see the effect on several different tissues of mice. In black, there is the typical viral load with no compound at all during the infection. And then we apply the compound, and the gray bars represent the reduction of the viral load thanks to novobiocin. So we can see some cases there is a 2 or 3 orders of magnitude reduction with the compound. So the compound was very efficient in vivo, and now we are trying to get the results. We want to advance in clinical trials to see if the compound is efficient in humans, of course, and also try to get a crystallographic structure of the key enzyme with this compound. Since this would give more insights about the real pose of the compound, and this would allow the development of new drugs in Zika virus. Here, there is the detail of the publication. You can see here that there are many authors contributing to this work, and this is very reasonable because there are many different expertises involved with this work. From our side, computational chemistry. From the side of University of Hong Kong, there are in vitro, in vivo studies. Then we filed the patent, and now we are negotiating the licensing of this patent with a pharma company from USA, and if everything goes well, we would like some clinical studies to start with this compound. So thanks a lot.